Welcome everyone, and thank you for joining us for today's webinar. My name is Zoe Jackson, and I'll be your host for today's presentation. Oxford Instruments Nanoanalysis is the leading provider of cutting-edge tools that enable materials characterization and sample manipulation at the nanometer scale. Today we'll be talking about Quantitative EDS Explained, how to achieve great results. I'm joined by today's speakers, Dr. Simon Burgess and Dr. Rosie Jones from Oxford Instruments Nanoanalysis, and Dr. Duncan Muir from the Cardiff University School of Earth Science and Environmental Sciences. Just a little housekeeping before we get started. You've all joined the webinar in listen-only mode. If you have any questions during the presentation, please type them into the question box in the control panel as we go along. The question box is located on the right side of your screen. All questions will be answered at the end of the presentation. Now, without further ado, I'll hand over to our first speaker.
Hello and welcome to you all. In this webinar, our subject is EDS processing and quantitative analysis. I will cover how an EDS spectrum is processed and quantitative analysis calculated. I will then hand over to Duncan Muir, who will talk about quant using standards in practice, its validation and some of its applications. I will come back briefly to describe how we calculate results using our standardless method, and then Rosie will describe the methodology needed to achieve great results and what accuracy you can expect. Rosie will then finish by talking about how we can go beyond EDS to analyze minor and trace elements, combining WDS with our EDS technique. In this first presentation, I will be opening up the lid on how spectra are processed and quantitative analysis done in our Aztec software, showing how our unique TrueQ technology can deliver a reliable and robust result when combined with good method and the technique my fellow speakers will talk about later. I find the best way to think about this is in two steps. Step one, how we take a spectrum and extract the X-ray intensities for each element, and then step two, how we calculate the quantitative result from those intensities. Step one delivers qualitative analysis, identifying the elements present using our auto element ID, assisting manual peak ID using functions like the fitted spectrum, and correcting for spectrum effects like peak overlaps, which will also give errors in X-ray maps, which can be corrected using our TrueMap software. Step two delivers quantitative analysis, whether using your own standards or our standardless method, to determine actual elemental composition. So let's start by opening up the box of technologies we use to extract X-ray intensities from an ED spectrum to provide qualitative analysis. There are 5 main stages to this process: correcting the energy scale, removing artifacts such as escape or pileup peaks, removing the X-ray background, fitting of element peak profiles to the spectral data, and finally extracting the intensities. I will be focusing on steps B to D, which I have highlighted in bold in this presentation. So firstly, a quick word about correcting the energy scale. It is critical that all the peaks appear at the energy we expect them to be for us to identify and process them. We calibrate the system on installation, and providing your room is stable, it rarely needs to be done again. You can redo this calibration using the energy calibration in the optimize step in Aztec. Minor changes are corrected in real time as our system constantly monitors and corrects for changes in the zero peak or zero strobe. It is worth highlighting this is not the peak you see near zero energy in your spectrum. This is what we call the triggered noise and should actually appear at an energy slightly above zero. The next stage is to remove or correct for any non-element artifacts. These are escape peaks and pileup peaks. Escape peaks are never visible in Aztec and are corrected automatically. Pileup peaks are caused when two X-rays arrive close enough together in time the system sees them as one X-ray event and counts them together. This is more likely the more X-rays are hitting the detector and therefore increase with and are much more obvious at high count rate. They are corrected if the pileup correction option is turned on in Aztec. Pileup not only produces artifacts, but also reduces the intensity of the element peaks. Therefore, to measure intensity of each peak accurately and ensure it does not change with count rate, it is important that the pileup is not only removed, but returned to its correct energy, as shown by the arrow here, where the pileup sum peak intensity in the uncorrected spectrum is returned in the corrected spectrum into the element peak, in this case the one at half the energy. The next stage is to remove the X-ray background or Bremsstrahlung. There are two common methods to do this. One is to calculate the background, fit it to the spectrum and subtract it. It is worth stressing we do not subtract backgrounds in Aztec. We believe this method is incompatible with the goals of microanalysis. It is operator dependent and with user interaction required for the best results, not compatible with instant analysis and results. In Aztec, we instead use the other method, where we filter out the background using a convolution filter called a top hat due to its shape. This is an operator independent and more robust approach. The effect of the top hat filter is to change the shape of the spectrum. The X-ray peaks are still there, and they have the same intensity when you add up the positive and negative parts of the resulting spectrum. But now the background has gone. For those of you who have heard of this term, this is the filtered parts of filtered least squares fitting method. To calculate the area of each of our X-ray peaks, we need to fit peak profile shapes for each element line series to them. To do this accurately, particularly where peaks overlap, we need extremely accurate profile shapes. So let us now look at how we have taken a library of line energies and intensities, as you can find in the literature, and turned them into the profile shapes we use, like the one shown here. We start with the library of lines and their energies and intensities. In this example, we show the ones for the nickel L line series, L alpha, L beta, etc. As I go through the following slides, note the subtle but important changes we make. Firstly, experimentally checking each line and making modifications where the intensities don't fit with those shown in the literature. Next, correcting for the change in our EDS detector's collection efficiency with energy. We will come back to energy efficiency later, but it's important to note all our detectors are tested on a SEM in our factory before they are shipped to ensure key performance parameters like efficiency will fit our software technology. Then we correct for changes due to the accelerating voltage. One of the reasons why a spectrum in Aztec must have the accelerating voltage saved with it to be processed is this. However, normally it is read by Aztec directly from the microscope. After that, we correct for the resolution of the detector, and we start to have something that looks like a peak rather than a set of lines. The resolution of every spectrum is monitored and corrected using the zero strobe measurement I mentioned earlier. Finally, in reality, peaks are not Gaussian, and we correct for the real shape of the peaks produced by our detectors. Again, this is something we check on every detector before it leaves our factory, but this is the kind of level of accuracy you need if you want the auto ID routine to look for all the elements that might be present in a sample. Once we have our profile, it goes through the same top hat filter to produce the same strange peak shapes as that of your spectrum when it's had the background removed. We then do the least squares fitting, a method to determine what profiles and in what proportion make up the spectrum being analysed. In this example, fitting the titanium K and barium L profiles to the spectrum containing overlap peaks of both, we can identify that they are present and determine the intensity due to each of the two elements. To be done accurately, this fitting method requires the peak energy and peak shape to match, with also all the artifacts corrected or removed. And that's it for step one. That's how our TrueQ technology algorithms take a spectrum and turn it into a spectrum with peaks automatically identified with their elements, with intensities allowing us to create the fitted spectrum shown as the pink overlay in the bottom spectrum, or to calculate a true map. So with these technologies, we can provide the tools to allow overlaps such as that between the titanium K and barium L to be correctly attributed in the spectrum to show that titanium and barium are present, and where titanium and barium are found in a sample, correctly using TrueMap. So the next step is step 2, quantitative analysis, where we take that set of lines and intensities and turn them into compositions. So what is inside that technology box in Aztec? Let's think first about quant analysis where you collect your own standards. Why would you need to do that? The fundamental equation for calculation of concentration of an unknown element is based on the K ratio of the intensity of the unknown to the intensity in a standard. Of course, we've covered how the intensities are calculated in step 1. We also need a matrix correction, here we call ZAF or ZAF, that corrects for the influence of chemistry of the material on the X-ray emission probability. One aim is often to use standards of a similar composition to those of the unknown sample, as this will keep the matrix correction and any error associated with it small. So why measure the K ratio and measure standards? To answer that question, let's start with the fundamental X-ray intensity equation shown here. This adds a whole load of additional parameters that need to be known, those listed in the bottom left, in addition to those which are in the quant calculation equation we just saw, which are listed in the orange box. However, if we form a K ratio measuring the intensity in the standard as well as in the unknown, we form this type of equation, and what you can see is that many of the terms are the same. And these terms cancel out, although it is worth saying only if they are the same, which in many cases requires consistent good experimental technique, a subject covered in the following presentations. With all the cancelled out terms, we actually get straight back to our equation for quantitative analysis. So EDS is a relative technique, comparing the intensity in an unknown to that of a standard of known composition. Then all you need to do is measure the intensities and worry about some additional parameters, the accelerating voltage, which as I mentioned earlier is saved with the spectrum, and the takeoff angle, which is known by the system and the software, but only if the sample is positioned at the correct height. One final point to note is that ZAF is a general term for matrix corrections, and to avoid any confusion, the one Aztec uses due to its proven accuracy for all elements is called XPP. So I'll now hand over to Duncan, who will talk about how he uses Aztec EDS to measure compositions with standards in practice. Over to you, Duncan.
Thank you, Simon. So I'm going to talk today about quantitative EDS, which really boils down to performing EDS on standards as well as our samples to assess our accuracy. I'm going to explain why we might want to do this. I'll go over briefly a typical method that we would use, and I'll finish by showing what the benefits are of putting in the extra time and effort to perform quantitative EDS by showing some data from recent publications from work we've done in our lab. So what is quantitative EDS? In simple terms, really any sort of quantitative analysis is a measurement performed in conjunction with standards, and in doing so we can validate the quality of our data. We measure standards really to determine the accuracy of our analyses, and when I say accuracy, that is meaning how closely a measurement conforms to a standard value, or you can think of it as the trueness of a measurement. With silicon drift detectors today, we're rarely counts limited when performing EDS, so our precision is typically pretty good. So in the images at the bottom of this slide, you can see in the first example, the precision is poor, there's a lot of scattering the data points. This is rarely the case with EDS, as I say, because we get such high count rates. Our precision tends to be fairly good, as demonstrated in the right-hand image here. But what we don't know is how accurate our measurements are. So when we are performing quantitative EDS, we are looking to try and achieve an accuracy or relative accuracy of our measurement of within plus or minus 2%. And it's only by measuring standards that we can confirm this. So what do we need to perform quantitative EDS? Well, we need, as always, if we're doing any sort of quantitative analysis by electron microbeam, we need a flat and polished sample, so typically polished down to a 1/4 micron finish. We need the sample to be conductive to electrons, so that means coating it typically with a thin layer of carbon. We need the sample surface to be orthogonal to the electron beam. We need relevant standards, and what I mean by that are materials which have a known chemical composition. We need to keep the SEM and the EDS settings the same between when measuring standards and the samples. And probably most importantly, we need a stable beam current. And this isn't always the case on an SEM, so it's always the key thing to check early on. The plot on the right-hand side shows a measurement of our beam current made over a period of about 30 minutes using our typical quantitative EDS beam setup. On this plot, time is on the x-axis and the beam current is on the y-axis. The red lines show plus or minus 1% relative change from the initial beam current measurement. And so we can see from this that there's very little variation in the beam current over this time period. What is our setup for quantitative EDS? Well, in Cardiff we use a Zeiss Sigma HD field emission gun SEM. We almost always do our quant work at 20 kV with a final aperture of 60 micron diameter, which gives us a beam current of around about 1.5 nanoamps. You can see in the image here that we have two large area EDS detectors on the SEM. We use these typically with a process time of 3 microseconds, with a dead time of around about 40%, which equates to output count rates of around about 100,000 counts per second. So when we count for 10 seconds, we end up with 1 million counts in a spectrum typically. The spectra are pulse pilot corrected. We work with totals that are not normalized to 100% when we're doing quant EDS. And for oxides, we calculate oxygen by stoichiometry. For non-oxides, such as sulfides, we extract all elements from the spectra itself. So I want to talk a little bit more about standards, reference standards. And you can see when we're performing quantitative EDS, we typically have a couple of blocks of reference standards, plus a separate mount with a cobalt for our beam measurements, and then some space for our samples as well. And really a reference standard is any material with a known composition, and typically the composition is agreed or verified by independent analyses from different labs. Standards can be pure elements such as metals, they can be alloys, they can be minerals and glasses, and these reference standards can be used in two slightly different ways. We can use them as secondary standards, which are chosen to assess the accuracy of our measurements. And when choosing a secondary standard, we basically want that material to be as close as possible in terms of composition and type of mineral or metal or whatever to the type of material we're measuring in our unknown or in our samples. We can also use the reference standards as calibration standards. So we would use these to calibrate the EDS system with the intention of increasing the accuracy of the measurements on a given element. And when choosing a calibration standard, we basically just want the element of interest to be present in relatively high concentration, so greater than 10 weight% is recommended. So having put our samples and reference standards, including our cobalt for beam measurement or copper for beam measurement, into the sample chamber and pumped down the SEM, typical workflow for doing quantitative EDS first involves measuring the beam current. So we'll use cobalt to measure the number of counts in the peak for cobalt, and we check this so a change in the beam current approximately every 30 minutes. We then measure a secondary standard to check the accuracy of each element within that measurement. And if we are measuring a glass, then we will use a glass secondary standard of a similar composition. So the table down at the bottom of the slide shows a composition of BCR2 in yellow, which is a basalt glass standard. Spectrum 1 in light blue is the measurement that we performed for our secondary standard. And then the very bottom row of that table is the relative deviation, or the accuracy if you like, the relative accuracy. And we can see that in black are elements which are within plus or minus 2%, and in red I've highlighted elements which are outside of that range. For the elements that are outside of the plus or minus 2%, we can improve or try to improve the accuracy of those measurements by calibrating or standardizing the EDS system. So that's the next step. We measure a standard which has greater than 10% of the element of interest, and having measured that spectrum, we use the peak from that spectrum to calibrate our EDS system to say, well, that peak equates to this much in terms of weight percent of that element. So if we take aluminium oxide as an example, you can see that it is 3% high in the analysis of spectrum 1 compared to the standard. If we measure, let's say, plagioclase as our calibration standard, plagioclase has a greater than 10% concentration of aluminium, and if we use the aluminium peak from plagioclase to calibrate our system and apply that new calibration to the very same spectrum number 1, measurement of BCR2 basalt glass, we can see that we've now improved our aluminium oxide measurement from plus 3% relative accuracy to minus 1%. And so we can repeat this procedure until we've maximized the accuracy on each of the elements of interest. Having done those checks and calibrations, we can then go on to measure our samples or our unknowns, confident that we're getting the best accuracy possible. So let's have a look at some secondary standard data that we've acquired on our system. On the left two plots is pyroxene data for the Kakanui augite. In the right two plots is from Astymex plagioclase. And in each of the plots, the standard value is the red square, our actual data, our quantitative EDS data in blue, and the shaded areas showing plus or minus 2% from the standard value. You can see that the majority of the secondary standard analyses that we've performed fall within that plus or minus 2% range. There is some creep over into 3% for calcium, for example, in the Kakanui augite. Some of the iron drops a little bit low, the augite also. And for the majority of the plagioclase data, which is averaged with standard deviation plotted, most of the data falls within that plus or minus 2% boundary.
Now, having demonstrated that it's possible to produce such high accuracy EDS data and to know what our accuracy actually is, the question still stands is, why bother? And a follow-up question to that I often get asked is whether our quantitative EDS data is publishable. And I'm happy to say that the answer is a definite yes from our experience. So various researchers using the lab in Cardiff have published their quantitative EDS data in a wide range of journals on a wide range of different scientific topics. The type of data required has ranged from analyses on glasses, silicate glasses, on a wide range of different silicate minerals, so olivine, pyroxenes, plagioclase, amphiboles, iron oxides, as well as sulfide minerals, which were measured in diamond inclusions. In Cardiff, we have quite a lot of researchers working on mid-ocean ridge processes, so looking at rocks that were recovered from the sea floor. And again, we've published a wide range of quantitative EDS data in various journals on this topic, and it's one of these journals that I'll be talking about today, which is a recent paper in Nature which looked at the magmatic system or the roots of volcanoes along the mid-ocean ridge in the Atlantic. And that's what I'll talk about a little bit more now. So in this paper, the researchers recovered rocks from the mid-ocean ridge in the North Atlantic, and their aim was to investigate the magma processes that were occurring beneath the seafloor prior to eruption in forming these volcanic rocks. For those unaware, the mid-ocean ridge is where new ocean crust is formed along the spreading axis of each of the Earth's ocean, and we can see it nicely demonstrated in this topographic image on the left-hand side. Prior to this study, and as demonstrated by the image on the right-hand side of this plot, it was thought mid-ocean ridge magmas crystallized no deeper than 5 or 6 kilometers below the seafloor, and hence that the magma crystallization pathway or history was fairly simple and straightforward. So researchers from Cardiff prepared these samples for investigation on the SEM. We were able to do all our backscatter imaging, element mapping, and quantitative EDS on one system. And these investigators were interested in melt inclusions, which are small pockets of melt trapped within a growing crystal. And once the melt is trapped, the crystal acts as a sort of pressure vessel, isolating this trapped pocket of melt from the external environment. Now, if we are able to measure the volatile content, so the carbon dioxide and water content of the trapped melt inclusion, because we know what the solubility of these gases are in silicate melts, we can start to estimate or calculate the crystallization depth of that crystal that trapped the melt inclusion. Now, previous studies have concentrated on olivine from mid-ocean ridge rocks, but these researchers went one step further and studied melt inclusions in plagioclase as well as olivine. Now, in order to measure water and CO2, one must first characterize both the host crystals and the melt inclusions themselves. So these researchers used quantitative EDS to measure the plagioclase and olivine chemical compositions, as well as the melt inclusions that were found within these crystals. Once they'd measured the compositions, the researchers then went on to use secondary ion mass spectrometry to measure water and CO2. And the actual quantitative EDS data in this case was used to calibrate the counts from the SIMS. So you can see in the bottom right hand side, this is quantitative EDS data for melt inclusions that were found in plagioclase in orange and in olivine in blue. And you can see that they share a relatively similar chemical space with a bit more variation in the melt inclusion composition from the olivine crystals. So hopefully this demonstrates that quantitative EDS is worthwhile technique pursuing. It uses standards to validate the accuracy of data acquired by EDS. In order to do that, we need flat, polished and conductive samples, as always with any electron microbeam quantitative analysis. We need a wide range of relevant standards for which we know the composition of. And importantly, we need a stable beam current, and we must be able to measure that. We've shown that major elements can be measured at 2% relative accuracy by calibrating the EDS system. And in doing so, it provides added value to EDS data, which then becomes publishable in a range of high-impact journals, and can also be used to calibrate trace element measurements, for example from SIMS or laser ablation ICP-MS. With that, I'll pass you back to Simon.
Thank you, Duncan. In this next section, I would like to reopen our quantitative analysis technology box to look at standardless analysis, or as we view it, what if the intensity of the standard is not measured by you, but it's measured by us in our factory, so you don't need to measure standards yourself. Let's go back to our K-ratio equation, but in this case where we measure the standard intensity in our factory. I have highlighted in blue all the factors specific to our factory measurement. In this case, fewer terms cancel out, and we are left with a K-ratio which has 4 more terms highlighted in yellow than that we saw when we were measuring our own standards. So what are these additional parameters? The first two are the beam current and the solid angle of the detector. These are typically removed in standardless analysis by forcing the analysis total to 100%. However, there is a better approach, which is to measure a single pure element sample. This is the beam current measurement found in the optimized navigator in Aztec. The acquisition time is known and saved with the spectrum. That leaves a parameter we met earlier, the detector efficiency. Typically modeled from theory, this was something we found to be too inaccurate, and therefore we have measured the efficiency directly using a synchrotron. By including the measurement of a pure element sample, we need to add terms for the intensity measurement of that element and the efficiency of the detector for that element. Having done that, the beam current and solid angle terms can be removed, and there is no need to normalize the total to 100%. This is something that only works if all elements are measured and can hide many errors that would be made obvious by a bad analysis total. All we need to know now is the efficiency of the detectors used at every element line measured, including those used to collect the standards in our factory and the one you will use to analyze your samples. The efficiency curve, like the one shown here, shows how many X-rays hitting the detector are actually measured at each X-ray energy. In this case, showing only the theoretical effect of the X-ray thin window, you see a much lower efficiency at low energies. To create efficiency curves for our detectors, we started by measuring a detector on a synchrotron. This allows us to create an X-ray beam of known intensity that can be monochromated to a single tunable energy. With this, we can see how much of that intensity we can detect at different energies. From that detector of known efficiency, we can create a new efficiency curve for each new detector type we introduce by careful comparative measurement of different materials on an SEM. In this example, we show the creation of the efficiency curve for our exact detectors. It is worth stressing again that all detectors are tested in our factory before shipment to check they match with the expected efficiency for that detector type. All that is left for us to measure is the intensity in our standards in our factory to create our factory standards database. As we only need to do this once, we can take and have taken great care over it using the procedure that we show here. And that's it. By making all these careful measurements on synchrotron and SEM, we are left with an equation where you have the same parameters to measure as in the analysis with standards we discussed before. A very similar K-ratio type equation. So as a user, instead of measuring the intensity in known standards for all the elements in your samples, all you need to do is measure the intensity in a single pure element standard, and then to measure your sample. We have covered the rest, such as the efficiency of your detector, another example of our TrueQ technology. I will now hand over to Rosie, who will present our recommended workflow and procedure for achieving good, accurate results and what accuracy you should expect using the standardless analysis approach.
Thank you, Simon. I'm now going to take you through our suggested best practice workflow for achieving accurate quantitative EDS results on your system with Oxford Instruments hardware and software. These first pointers relate to sample preparation and stage setup. Firstly, as Duncan said, samples and any standards being used should be flat, polished and conductive. If a sample is not flat, the EDS might be affected by shadowing, where X-rays are blocked from reaching the EDS detector due to higher parts of the sample. Topography on the sample can also result in an incorrectly calculated X-ray absorption correction. This is especially critical for light elements which are much more heavily absorbed by the sample. If the sample itself is not conductive, then charging can result in the landing energy being significantly less than the beam energy, which can drastically change the intensities of the X-rays emitted from different elements in the sample. Therefore, a conductive coating should be applied to stop the sample from charging under the electron beam. Typically, a carbon coating of between 10 and 20 nanometers in thickness is sufficient to make most sample types conductive. If you wish to obtain unnormalized quantitative EDS results, then in addition to the sample or the unknown that you wish to measure, you will also need a beam measurement element. This needs to be an oxide-free pure element such as copper, cobalt, manganese or silicon, and the same rules apply to this beam measurement element as for the sample. It needs to be flat and conductive. If you don't have a suitable pure element standard, then something like some copper tape, usually used to stop samples charging, could be used for this purpose, although it's not totally ideal. If we go back to the equations that Simon presented, the reason that the beam measurement element is needed is that it's important for determining the beam current and the solid angle for the unknowns, so that these can be compared with those for the measured standard. Once prepared, both the sample and the pure metal for the beam measurement should be loaded into the SEM at the same time, and ideally so that they are at the same height on the SEM stage. Once the sample and the beam measurement element are loaded into the SEM, the EDS detector should be fully inserted so that it sits up against the stop nuts, as shown in the figure on the right. The sample surface should also be set at the recommended working distance for the system. If you don't know what the recommended working distance for EDS analysis on your system is, then there are a number of places in the Aztec software that you can find out. The most obvious place is the EDS rate meter located in the menu view, as you can see in the figure on the top right hand side of this slide, and there is a value displayed for the recommended working distance. To set this up, set the acceleration voltage to what you think you want to work with, and remove any lens hysteresis. Then set the beam focus at the recommended working distance, and use the stage Z position to bring the sample into focus at the recommended working distance. This process is being shown in the video on this slide. The reason this is important is that the sample height is critical for determining and maintaining the correct takeoff angle, and again I highlight that this is used in the key equation that Simon presented. Bear in mind that every time you want to acquire an accurate quantitative analysis, the sample should be carefully focused at the correct working distance. So if your sample changes in height or you are analysing multiple samples, then the sample must be refocused by adjusting the Z height of the stage before analysis at each point. Once the EDS detector and sample are set up in the correct positions for analysis, the analytical conditions on the SEM can be set. Firstly, adjust the accelerating voltage. 20 kV is normally a good starting point, as this will excite a quantifiable X-ray line for all elements. The beam current should then be set to give a suitable EDS input count rate. Too few counts will produce bad counting statistics and errors, and too high a count rate will produce lots of pulse pilot peaks and a high detector dead time. With the beam position on the sample, adjust the beam current so that the input count rate is somewhere between 50 and 200,000 counts per second. This is a good starting point for most modern silicon drift detectors, although we have shown that good quant EDS can be produced for count rates as high as 400,000 counts per second on the Ultim Max EDS detectors. The process time should be adjusted to achieve the best spectral resolution while keeping the dead time below 50% for a given input count rate. We recommend using process times of between 3 and 6 for quantitative EDS analysis. These process times have been fully tested to give quantitative results with count rates as high as 400,000 counts per second. For a given process time and input count rate, ensure that the detected dead time shown in the EDS rate meter is 50% or below. With our latest pulse processor, there is no requirement to be at particularly low dead time, and 50% dead time is actually when counting is most efficient for the selected process time. If you do find the dead time is over 50% for a certain count rate, a lower process time can be selected, although as I said, a process time of lower than 3 is not recommended for quantitative analysis, as there is likely to be significant pulse pileup. That said, I should also point out that process times of less than 3 can be used where quantitative accuracy is not so important, for example in EDS mapping or feature analysis. When making quantitative measurements, it's important that the beam is stable, particularly between the beam measurement and the unknown sample analysis. Duncan also highlighted this in his presentation. Ideally, the beam current should not fluctuate more than 1% over 5 to 10 minutes. One way of checking this is to make a number of beam measurements, which I shall explain on the next slide, over the course of 5 to 10 minutes. The result of each beam measurement gives a percentage change since the last beam measurement, as circled in the image on the right. Finally, make sure that the field of view is smaller than 0.5 mm to avoid any occlusion by the collimator of the EDS detector. If you want to produce unnormalized results, then a beam measurement is required to calculate the analytical total. In the Aztec software, this can be done in the Optimize Calibrate step by selecting the beam measurement option and an appropriate pure element from the drop-down list. As with this sample, make sure that the pure element is in focus at the correct working distance for the system by adjusting the stage Z position. Once in focus, set the magnification of the SEM to 10,000 times or more, so that the measurement is being performed on a small spot on the pure element, and then press the Start button to make the beam measurement. An example is shown here for cobalt. This spectrum contains 600,000 counts and took less than 20 seconds to acquire. Once the beam measurement has been made, you can go on to collect unnormalized quant data from unknown samples, but bear in mind that if the accelerating voltage or the beam current are changed, then another beam measurement will be needed before any further unknowns are analyzed. It is also recommended best practice to regularly perform beam measurements throughout the analytical session, just in case there is drift over the course of the unknown measurements. Before measuring the sample, ensure that appropriate coating information is added, both for the sample and the beam measurement element. This can be done in the Describe Specimen step in Aztec. Once located on the sample, the electron beam should either be in spot mode or scanning over a very small region. Or alternatively, a high magnification on the SEM can be used, which is the equivalent to being in spot mode or scanning over a small area. Which of these beam options is most appropriate will depend on the EDS Aztec navigator that you are in, for example, whether it be Analyzer or Point and ID. But the key message is, analyzing a very small and well-chosen point on the sample is important for quantitative EDS, as the matrix correction applied requires the sample to be homogeneous within the interaction volume of the electron beam. We then come to the settings for the EDS spectrum acquisition. As a starter, we would recommend adjusting the EDS acquire spectrum settings to collect an EDS spectrum containing 1 million counts. Also ensure that the pulse pilot correction is turned on, and this will automatically remove those pulse pilot peaks that Simon spoke about earlier. Quantification will not work well, and EDS will never yield accurate results if the elements present in the sample are not correctly identified. So during or after the EDS spectrum acquisition, it's important to verify the elements that have been automatically identified by the auto ID. This can be done by assessing the fitted spectrum, shown in pink on these figures, the simulated theoretical spectrum, which is usually shown in light blue, and the analytical total. The figure on the bottom left of this slide shows an example where, when comparing the collected spectrum with the fitted spectrum, we can see that there is a mismatch, giving a good indication that an element might be missing and wasn't picked up by the auto ID. Once calcium has been manually added, as shown in the middle figure, a much better match is observed between the acquired and the fitted spectra. The Candidate Elements tool can be used to identify potential elements in regions of the spectrum where a mismatch exists, as shown in the figure on the bottom right. Once collected, the quantitative EDS results can be viewed and interrogated in the Calculate Composition step in Aztec. It is a good idea to check the quant settings that are being used to calculate the result. For example, do you want to calculate the quantitative weight percent for all elements, or perhaps it would be preferable to calculate the oxygen by stoichiometry, as may be the case for geological minerals. There is also the option to normalize the result, which normalizes the result to give an analytical total of 100%. If you've performed a beam measurement, then the results can be unnormalized. An unnormalized total is often a good first check of the analysis quality. If the analytical total is more than plus or minus 2% of 100%, then it is possible that an element is missing from the analysis, or that the analysis has been conducted on a bad spot, or possibly that the beam current has changed since the beam measurement was made. However, you should bear in mind that for some samples it may be usual to obtain totals that are less than 100%, for example, hydrous minerals. Finally, check that the appropriate quant standardization database is being used to calculate the quant result. There are a choice of factory quant standardizations, which is basically the standardless approach that Simon spoke about, Or you can acquire your own standardizations, and these will appear in the user drop-down list. And this is the methodology that Duncan spoke about, where they measure their own calibration standards for quantitative EDS. And that concludes our best practice workflow. In this next section, I shall present what level of quantitative accuracy can be achieved with an Oxford Instruments EDS system and the Aztec software if you follow our best practice workflow and what we have referred to as the standardless approach. We have assessed the accuracy of our standardless approach by performing a series of experiments using a standard block containing many commonly occurring elements. In this presentation, I'll show focus on the quantification of elements with atomic numbers higher than sodium. The elements that were measured are highlighted here in the periodic table. Aztec layer probe was used to measure the thickness of the carbon coat on the standard block, and the coat was found to be 13.5 nanometers thick. A beam calibration was performed on cobalt after every 5 acquisitions to measure the beam current and ensure its stability. Measurements were made using an Ultim Max 100 EDS detector mounted on a Leo SEM. The quantified elements were automatically identified by the Auto ID. The collected spectra were corrected for pulse pileups, and all elements were quantified by direct measurement, with the exception of oxygen contents in oxides, which were calculated by stoichiometry. All results reported here are unnormalized, as our standardless approach allows the calculation of a true analytical total. 3 sets of results are shown here at 2 acceleration voltages, 20 and 15 kV, and 2 input count rates, 50 and 200,000 counts per second. The plots show the measured weight percent compared to the nominal concentration for the standards as a ratio. A value close to 1 indicates a good agreement. The average and the relative standard deviation are shown on each of the plots. We demonstrate that there's a good agreement between the measured and the nominal concentrations at both accelerating voltages and input count rates. The relative standard deviations approach a value of 2%, which is usually quoted as the level of accuracy that can be achieved by an electron probe, and is also the value that Duncan was aiming to achieve using his standards-based approach. Based on this evidence, along with other work that we have done, we believe an accuracy of 5% or less is achievable on an Oxford Instruments EDS system using our standardless approach if you follow our best practice workflow. So can we go further and extend the capacity of an analytical SEM? In this final section, I'm going to talk about how we can take quantitative composition analysis in an SEM a step further by adding wavelength dispersive spectrometry for trace element analysis. Wavelength dispersive spectrometry has a higher spectral resolution and a higher peak to background ratio in comparison to EDS. This means that adding a WDS spectrometer to an SEM can expand the analytical capability to resolve peak overlaps that might exist in the EDS spectrum, and to accurately measure trace elements down to a sub 100 ppm level. The Oxford Instruments wave spectrometer is the only WDS solution for the SEM with true electron microprobe WDS spectral resolution. This brings the capability to fully resolve all of the common peak overlaps, including all of the transition elements. We now have WDS fully integrated into the Aztec software platform, and Aztec Wave can be used to make quantitative compositional measurements using combined EDS and WDS. Aztec Wave enables EDS to be used to measure the major elements present in the sample, and WDS to be utilized where it is needed most, to either measure minor or trace elements, or to measure elements where a peak overlap exists in the EDS spectrum. These are some example results obtained using combined EDS-WDS analysis in Aztec Wave. Sticking with the geological theme, these are some results obtained from olivine minerals present in a gabbro. The table shows average compositional data from exactly the same 8 points on the sample using different microanalytical techniques. The top row shows published results obtained using WDS on an electron microprobe, and the rows titled SEM WDS only and SEM EDS plus WDS show the data obtained using SEM-based WDS and Aztec Wave. In all cases, the oxygen is calculated by stoichiometry, and the analytical totals are shown as normalized. If I draw your attention to the bottom 2 rows, these show the results where the major elements have been analyzed via EDS, and the minor and trace elements, highlighted with the orange boxes, have been measured via WDS. As you can see, the results obtained are with an error of those obtained using the electron microprobe, and that the calcium has been measured down to a level of 100 ppm using SEM-based WDS. And finally, this slide concludes our presentation. So reiterate what Simon outlined at the start. These are the key things that we hope you will take away from this webinar. We have shown that if recommended best practice is followed, that publishable quality EDS data can be obtained and valid scientific conclusions can be made. We have highlighted that system validation is important and should be used to demonstrate the quality of results that your particular SEM EDS system can achieve. For example, this can be done by measuring secondary standards and comparing the measured and published results. We hope that you've understood something about our TrueQ technology, what it does and why it's important. We have also shown that what we call standardless EDS analysis in Aztec can produce good quantitative results for many applications. And for those of you who want to push your system further to measure trace elements, WDS can be added to the SEM to bring detection limits down to the sub 100 ppm range for many elements. So thank you again for joining us for this webinar, and we hope that you find it informative and useful.
Thank you, Rosie, and thank you to all of our presenters today. We'll now go ahead and take some time for questions. It looks like we've already had a number that have come through during the presentation, so just a reminder to please type your questions into the question box in your control panel to the right. Now for our first question, Simon, one of your earlier examples, sulfur is showing in red. Why is this so, and how can we make it normal?
Oh, I think this is the mini quant on the title slide at the start. So the element at the bottom, sulfur, was in red. This is about the uncertainty of the automatic element identification. So eventually there comes a point at which the statistics for the automatic identification algorithm become bad. And at that point, the default behavior of Aztec is to highlight those elements in red. And that's really a signal for the analyst to go and use some of our manual tools, such as the fitted spectrum, to check that the element is really present. If you don't like that behavior, it can be turned on and off. You need to go to the preferences in the tools menu, select the EDS spectrum viewer tab, and look at the bottom for the highlight elements with poor statistics. And there you can turn it on and off.
Great, thanks, Simon. That sounds like a good feature. Now a question for Rosie. Can you quantify with a high degree of accuracy the amount of titanium and barium in a sample?
Hi, thanks for the question. Yeah, so titanium and barium have X-ray peaks that overlap each other in the EDS spectrum, which makes them slightly more difficult to measure using EDS. The EDS will quantify them, but this is an example where if you really want to accurately determine the concentrations of titanium and barium, that's an example where the WDS would really increase the capability of your system, and that's because WDS has a increased energy resolution in comparison to EDS, and it will be able to separate those titanium and barium peaks and produce a more accurate quantitative result for them.
Great, thank you, Rosie. Now I believe this question is for Duncan. And for how long are the standards of the standard block trustable and stable?
Yeah, so the standard block will, depending on what materials are in your standards, they should remain stable for decades, if not longer. Certainly for geological materials, we'll be talking millennium if you're going to keep it in your lab that long. But there's two things to consider. One is that the carbon coat will degrade over time. So in our lab, we tend to repolish the standard blocks approximately once a year just to remove and refresh the carbon coat. The other thing to consider is that certain elements can be mobile under the electron beam. So, for example, in geological materials, sodium is mobile under the beam. So in glasses or in plagioclase, sodium will migrate away from the beam during an analysis. And this can redistribute the sodium in the standard. So it's probably good practice to repolish to try and expose a fresh surface of your standard approximately every year or something like that if you're able to. But we find that our standards stay stable indefinitely, essentially. So.
Oh, well, thank you for that and good tips. Now I think we've got a question for Simon, which is how does the EDS detector position in terms of working distance affect quantification in relations with takeoff angle?
So I think it's worth saying that what we've shown today is the method that we use to ensure that we achieve the best possible accuracy. And the more effort you put into it, the more accuracy you will achieve. And essentially, the main reason, certainly for major elements, why WDS is perceived to be a better technique and more accurate is that the level of care taken over the analysis and the level of procedure and process used is much more rigorous. So let's consider the working distance as an example. As Rosie said, the important thing is you need to match the working distance to the recommended one for the detector. And if it's not at that correct working distance, then what is changing is the takeoff angle. If the working distance is shorter, the takeoff angle will be lower than our algorithm would expect. And if the working distance is longer, then the takeoff angle will be higher. And that will change the interaction of the X-rays with the sample. So a common thing to worry about is absorption of X-rays in the sample and its effect on the matrix correction. If the absorption in the sample is very high, which we commonly see with low energy X-rays, so those used when you're analyzing light elements, if the working distance is too short, then we will overestimate the absorption and we will calculate a concentration that's too low. And again, vice versa, if the working distance is too long, the absorption will be underestimated and the concentration will be too high. And we usually find for problems, for example, for oxygen analysis, the most common problem causing inaccuracy is that the working distance is not set to the correct height. The care you take and the amount of this procedure that you follow will affect the accuracy of the results that you can achieve, because you will be getting closer and closer to the algorithm that we are using to calculate the quantitative results.
Great, thank you for that, Simon. Now, Rosie, is it possible to increase the accuracy by measuring multiple areas in a uniform sample and averaging over them?
Yeah, thanks again for the question. So, as I said in my presentation, what's important to produce good quantitative EDS is that the interaction volume in your sample is homogeneous. So if you're analysing larger areas and multiple areas on a sample, you really have to be certain that it's going to be uniform and that you're not going to hit anything that is heterogeneous, because that will affect the accuracy of your quant results. Yeah, so if you want to increase the precision on the analysis, so to collect more counts in the EDS spectrum, which will help your precision, then perhaps counting for longer on a single area would help in the precision. Thanks.
Great, and I think we have time for just one more question. So how far is EDS comparable with WDS? Which is better?
Yep, so that was something I came to towards the end of my presentation. It really depends what you want to do. So we've shown here that EDS can produce good accurate quantitative data for like major to minor elements. But if you really want to push your system to measure elements at the trace element level, so in the sort of 100 ppm level and below, then that's what adding WDS to your system can bring. Additionally, if you're looking at samples where you have issues in your EDS spectrum with overlapping X-ray peaks, which the EDS spectrum finds it difficult to separate those peaks, then adding WDS to the system, because it has increased resolution, it's possible to separate out some of those common peak overlaps and therefore produce more accurate quantitative results.
Amazing. Thanks so much, Rosie. And thank you to all of our presenters again today. So that's all we have time for now. So thank you everyone for your questions, and we'll get back to any of the unanswered ones individually. We really appreciate you coming. And a recording of this webinar will be available within the next week. So please be sure to check out the website and get in touch if you have any further questions. Thanks again for joining, and we will see you next time.